Hi, welcome back. I'm Andrew Penn, clinical professor at UCSF, and this is going to be a three-part module on rapidly acting antidepressants, looking at both GABA and glutamate, and also looking at an assortment of various approaches to anxiety, benzodiazepines being one of them, being a GABAergic treatment modality and also alternatives to benzodiazepines. So this first part here is going to be the segment on glutamatergic drugs. So we're going to be extending our learning about treatments for mood disorders and also focusing on treatment of anxiety as well. So as we recall from the STAR-D study, which largely was using monoamine approaches to treating depression, we left at least about a third of people still not well after 4 different layers of treatment. So clearly there's a lot of room for improvement in our current treatments. And fortunately, really for the first time in about 25 years, the advances with rapidly acting antidepressants offers some really new innovation and new hope for our treatments suffering with depression. So what we're going to be talking about in these modules here are these rapidly acting agents. So that's really what kind of draws them all together, whereas the monoamine approaching the drugs that affect monoamines directly are more likely to be slow acting. These are things that are working on GABA, glutamate, and then things that are working for sleep, such as melatonin and orexin, and also things for anxiety, such as histamine approaches, cannabinoids, and also the GABA drugs that work on anxiety, known as benzodiazepines. So as we talked about in the monoamine lecture, we mostly focused on the left side of this family tree. And we're going to circle back to some residual monoamines, so the histamines, antihistamine drugs. And then, as you recall, melatonin is a derivative of serotonin. So we're going to be talking a little bit about that. But most of today, we're going to be talking about the amino acid side of the family. So these are the drugs that work on glutamate and drugs that work on GABA and also benzodiazepines. So just as a review, you remember that glutamate is the most uh, common excitatory neurotransmitter in the brain. So we can really think of this as the gas pedal. And there are 2 types of receptors for glutamate, 2 large families of receptor types. There's the ionotropic glutamate receptors. Those are those funnels that allow for ions to enter in from the outside of the cell to the inside of the cell, typically positively charged ions. And so those are the ionotropic receptors, such as NMDA and AMPA and kainate, which we're not going to spend any time talking about today. And then there are the metabotropic glutamate receptors. These are referred to as mGlu, M for metabotropic, and there's 8 different types of those. We're going to mostly focus on NMDA and AMPA today, and talk about how activating those has a downstream effect of creating synaptogenesis. So this is sometimes referred to as a tripartite uh, synapse. So, and the reason why it's tripartite, 3 parts, is that there's the typical presynaptic neuron and the postsynaptic neuron for glutamate. But then you'll notice that green blob to the right, that's an astrocyte. So astrocytes, we're going to talk more about in a moment, but this is critical for recycling the glutamate in the synapse. So we have our ionotropic receptors, as we mentioned, NMDA and AMPA. We also have the metabotropic glutamate receptors, and we also have this glutamate reuptake. And so that is accomplished through these excitatory amino acid transporters, EAA1 and 2, which draw the glutamate from the synapse into the astrocyte, where it is then cycled back through. It's broken down by glutaminase and turned into glutamine, and then repackaged for use in the cell as glutamate. Now, ketamine has really made a big impact on psychiatry in the last number of years. This is a drug that's been around a lot longer than that. It was actually FDA approved in 1970 as an anesthetic, and it got sort of test driven in the Vietnam War because, like morphine, it was a drug that could be carried by soldiers to give a buddy if they were injured and to assist in their evacuation. So it got a foothold in emergency rooms and as a surgical anesthetic because it works quickly. And the nice thing about it is it doesn't require you to manage the patient's airway. The patient can manage their own airway when they are under ketamine anesthesia. And so that's why it's useful in the field. Now that's at doses at, for anesthetic uses, maybe 2, 3, 4 milligrams per kilogram. The drug can actually be antidepressant as low as 0.5 milligrams per kilogram of body weight. And so we're talking about doses that are much smaller than those which are used in the emergency room. And this was an accidental finding. So back in 2000, this paper was published from Robert Berman at Yale, who was working on creating a model for studying psychosis and looking at ketamine as a possible candidate for that, and found that the model didn't work very well. But what was an interesting finding was that there was this rapid drop in depression scores in patients that had depression that happened in the course of maybe 4 or 5 hours or no more than a day. So very unexpected, interesting finding. It was replicated in a number of studies. And what you can see is that there's this drop in depression scores compared to placebo, but unfortunately, it tends to kind of crawl back up after a few days. Now, different people will get more mileage out of this. So some people can get several weeks of mileage before they need another treatment. Other people, maybe a week or so. So this is one of the biggest challenges ketamine as an antidepressant is that it doesn't have the kind of enduring effects that we would like to see, but it does have this very rapidly acting antidepressant effect. And this has been replicated in many different studies, and this is a meta-analysis showing that the antidepressant effects of ketamine in regular depression and treatment-resistant depression, or what we might think of as monoamine unresponsive depression, is pretty impressive. So, we definitely this appears to have a fairly reliable antidepressant effect. And to understand how this works, we need to understand a few different things. One of them is glutamate, and the other is astrocytes, and how astrocytes play a role in glutamate and possibly in depression pathogenesis. Now, when we think about the brain, we often think just about neurons, and the glial cells don't get a lot of attention until they mutate and become cancerous. So when we think of, say, a glioblastoma or astrocytoma, those are um, those are cancerous growths that emerge from these glial cells. But glial cells do a lot of different things. And the astrocytes are particularly busy. In fact, they're the most abundant glial cell in the brain. And they are part of what creates that blood-brain barrier. If you remember back to the first or second week, we talked about how these projections from astrocytes wrap around the capillaries in the brain and create the blood-brain barrier. They're also involved in the regulation of energy, oxygen, and glucose for those very active neurons. And as we'll see in the case of glutamate, help to regulate neurotransmission. So I think we're going to see a lot more about astrocytes in the years ahead. I think they've been kind of neglected in psychiatry, and I think there's a lot to be discovered there. So here's just again, another overview of the sort of glutamatergic system. So again, pointing out that presynaptic and postsynaptic glutamate. Glutamate is released from the presynaptic, comes across into the synapse. In addition, there is another transmitter here called glycine. And glycine is often thought of as a co-transporter, and we'll say more about what that means when we get to NMDA receptors. And you'll notice also that that astrocyte is there with those similar to kind of transporter pumps, the EAATs, and is pulling up that glutamate, transferring it back into the presynaptic neuron. I also want to draw your attention to the upper left corner here, where there's a GABA interneuron, and that's going to come into play in a moment. So we've really got these sort of almost 4 parts, really. So we've got the presynaptic and postsynaptic glutamate receptors. We have the GABA interneuron, which is synapsing onto the glutamate presynaptic neuron, and we have the astrocyte, which is recycling that glutamate. So let's take a closer look at the AMPA receptor. Now, Before we get too much into the AMPA receptor, it's important to understand how AMPA and NMDA work kind of in concert with each other. So they both are sensitive to glutamate. NMDA needs a second neurotransmitter called glycine. But let's focus on how these two work together for the time being. They both use glutamate as their neurotransmitter. You'll notice they're both these ionotropic um, uh, channels, right? So when they open up, these positively charged ions can flow in, but they work in this sort of two-part concert. So AMPA is going to open, it's going to begin to depolarize the cell. That's going to flow down the membrane to the NMDA receptor, which only when that current arrives can that fully open. So let's take a look at what I mean by this. So let's follow along on the left side for right now. So we've got glutamate is going to, oh, sorry, computer's being weird. Let's try this one more time. Appreciate your patience. So there's glutamate activating that those ion channels. Okay, and you see that sodium flowing in, calcium flowing in, allowing the cell to depolarize. So let's take another look at this here. Okay, so hopefully I've solved that technical problem here. But let's go ahead and take a closer look at this. So there's that glutamate coming in, sodium's flowing in. Now watch the NMDA receptor. There's a molecule of magnesium that's sitting in that channel. And it's not until the AMPA receptor begins to depolarize the membrane that that magnesium channel can get removed from the NMDA receptor. As long as that magnesium molecule is there, the NMDA receptor cannot open up and allow those positively charged sodium and calcium ions in. So this is how these two sort of they sort of work together. But let's go ahead and take one more look at this whole process. Okay, so we've got glutamate comes in, activates AMPA. AMPA allows sodium to flow in, begins to depolarize. Magnesium moves out of the way. Now sodium and calcium are going to flow in. So these two work kind of like they've got this sort of back and forth action that's happening as the signal is propagating down the line of the neuronal membrane. So we can kind of think of this as they're happening as a sort of 1-2-1-2 process here. So let's take a close look at that AMPA receptor. So AMPA is activated by glutamate. You see it's sitting in the cell membrane. So it's that sort of opening, that pore that opens up and allows those ions to flow in. So when glutamate attaches to it, sodium and calcium can flow into that AMPA receptor. We call that AMPA throughput. Sometimes you see that in the literature. So when this happens, it contributes to the cell depolarizing. Now, right next door is the glutamate receptor, NMDA receptor. Now you see this has two different neurotransmitters that have to attach before this can open up. So three things have to happen. Glycine has to attach, glutamate has to attach, and that electrical signal needs to be coming down the line to make that magnesium molecule that's sitting there. I've depicted that as like a manhole cover over this opening. That has to get out of the way. And that happens when that electrical charge comes down the line. So let's go ahead and take a look at this. We have glycine and glutamate have attached. That calcium is just waiting for its chance to come inside the cell and begin that depolarization process. Now comes that electrical current flowing down the cell membrane. Magnesium gets out of the way. And now that calcium can flow in, causing or contributing to the cell depolarizing. Now, where does ketamine fit into this? So ketamine sits on something called the PCP site, which is actually inside of that channel as it goes through the cell membrane. And it's called the PCP site because the precursor to ketamine was phenylcyclidine, or PCP, which is a drug better known for its sort of street use than it is its surgical anesthetic history. But the reality is that ketamine was actually created to try and improve upon PCP. So PCP, phenylcyclidine, was a surgical anesthetic when it was first created. And so ketamine actually attaches to the same site as phenylcyclidine, right there in the middle of that pore. And so when that is attached there, now calcium can't flow through and the cell does not depolarize. So that's sort of part one of the story. Um, I'm going to take a quick diversion here to talk about glutamate, astrocytes, and depression, and then we'll circle back to how these receptor activities contribute to a intracellular process which results in the cell growing, branching, and making new dendrites, something we call synaptogenesis, and how that contributes to an antidepressant effect. So we do know that glial cells are affected by elevated cortisol. I think the elevated cortisol is definitely a feature of early life adversity. We certainly see this in animal models. When you make a rat's life stressful, it increases their cortisol level, and that decreases their density of astrocytes. And in fact, these may be the early symptoms of depression, early signs of depression, I should say. And as those astrocytes are impaired, it makes it more difficult for the organism to remove that glutamate from those synapses. So we essentially end up like these neurons are kind of soaking in glutamate, right? They're not able to remove the glutamate from the synapse as well because they've got an impairment in the activity of the astrocytes. And so in animal models, we see this when you make a lab animal's life chronically but unpredictably stressful by leaving the lights on when it wants it to be dark, or turning the cage on its side, or any number of things that you can do in a chronic unpredictable stress model. What you see is in the brain tissue of that animal, you have less density of these glial cells. So those dark stained areas are glial cells, they're astrocytes. And we see a similar phenomenon in humans. Obviously, you can't ethically impose that onto a human subject, but looking at somebody who had major depression and died, they had fewer of these glial cells than a control subject. And this reduction in density is particularly noticeable in areas of the brain that are associated with depression, such as the dorsolateral prefrontal cortex and the amygdala and such. So this is kind of an anti-neuroplasticity, if you will. Okay, so with those astrocytes being impaired, then what we end up with is additional glutamate in the synapse. And when that happens, you get this thing happening at the bottom here. I want to point this out. These extrasynaptic glutamate receptors. So these are extrasynaptic. They're outside of the synapse, and they are a phenomenon that occurs when you have increased what's called tonic levels. Remember, we talked about this with norepinephrine, that there are tonic levels of glutamate. There's tonic firing of noradrenergic cells, and then there's phasic. So you can have a big burst, that's phasic, and the baseline amount of activity is the tonic level. So in this case, in a sort of depression pathophysiology model, we may see increased levels of glutamate. So this is kind of an excitotoxic state, as it's sometimes referred to. So we come up with this sort of pathway here where, maybe somebody is born with a certain degree of vulnerability to depression, encounters early life stress. Perhaps they have deficient factors for growing neurons, and that this state of being in a chronic state of stress increases glucocorticoids, causes cells to die earlier than maybe they should. We end up with fewer glial cells. With fewer glial cells, we're less able to clear that glutamate out. We get in this kind of excitoneurotoxicity state, and that perhaps this is what selects against cell growth or cells dying earlier than they should. If you remember nothing else from this talk, remember this. So ketamine is a rapid promoter of neuroplasticity. And by neuroplasticity, I mean this branching and dendritic synaptic growth from a neuron. So we can see this in this rat stained model here. This is the control, and then the bottom is sort of like, we go from like a tree in winter to a tree in the middle of spring, starting to branch out and really kind of push out new growth. So there's a lot happening in this diagram. These kind of diagrams I find incredibly intimidating. But if you take your time with them and you start to understand how to look at them, and often the best way to understand that is to read the caption that comes along with them, because they will often kind of walk you through the sequence of events. But I want to give you a high level sort of preview of what I'm going to talk about in the next few minutes, and then we'll circle back to this diagram and look at it in more detail. But here's sort of the top level read. So the end of the story I'm going to give away now. You grow dendrites and synapses. Okay, that's the bottom line. So everything is kind of funneling down to that point. So you see the synaptogenesis. That's what's illustrated here in this photograph. But what happens first is we get this ketamine, we get the inhibition of the ketamine neuron turned off. So what do I mean by that? So remember, we have these GABA interneurons, and remember, GABA is an inhibitory neurotransmitter system. So when when that GABA interneuron is influencing the glutamate neuron, it's preventing the glutamate neuron from firing and releasing glutamate. Now you'll notice here that there is a glutamate receptor on the GABA interneuron. If you remember, ketamine blocks these. So by inactivating the GABA interneuron, we're essentially kind of cutting the brake cable. We're preventing the GABA interneuron from hitting the brakes on that presynaptic glutamate neuron. What happens then? We get a big release of glutamate. So now you might be thinking, well, wait a second, isn't that bad? Like, I thought too much glutamate was excitotoxic. And you're right. Chronic tonic states of glutamate are not good. So it's kind of like having a lot of standing water sitting around. Like, you don't really want that. It breeds mosquitoes and disease. But this is like a big thunderstorm comes out and like washes all that out. Okay. So we get this big glutamate release. Okay. That's the next thing. That's the first thing that happens. And then that glutamate's going to go across the synapse and start activating these AMPA receptors. When AMPA receptors are started, they begin this glutamatergic cascade, something called BDNF, which leads to the activation of something called mTOR. So we'll come back to that in a second. But before that, on this right side of the slide, you'll also see that ketamine, when it's blocked, activates these growth cascades. So things that contribute to synaptogenesis. Get more into that in a moment. That extra BDNF that is being generated by the activation of AMPA receptors. So I want you to pay attention to these little yellow diamonds here. That's BDNF, and it's starting to activate these TrkB receptors. And you see TrkB receptors have this straight through line here to something called mTOR. And mTOR is one of the final common pathways for growing new synapses. And then finally, remember I talked about those extrasynaptic receptors that are outside of the synapse. So the ketamine will also turn those off. And why is that useful? Because those extrasynaptic receptors actually, when they are active, they inhibit mTOR. So we're kind of cutting the brakes on their ability to slow down mTOR by blocking them with ketamine. So all of this is going to funnel down into a synaptogenic process here. So let's get more into the details of that, and then we'll circle back to this because it'll make more sense. So ketamine is going to block the NMDA receptor, as you can see here. That's going to deactivate something called EFF2 kinase, and that begins a cellular process which creates BDNF. So BDNF, brain-derived neurotrophic factor, or kind of brain fertilizer, is going to start being produced. When that happens, the BDNF will activate TrkB receptors, as we saw in the previous diagram, and that's going to lead to increased synaptic growth. By the way, the EFF2 kinase is another area of potential interest as an antidepressant target. So keep your eyes on that one. The next pathway that's going to contribute to cell growth is something called mTOR. mTOR stands for mammalian target of rapamycin. And when NMDA receptors are blocked with ketamine, you get increased signaling in mTOR through that pathway that I showed you in the more complex diagram. And that begins a process, which we'll dig into, that's inside this black box. But on the outside of that black box is an increase in synaptic growth. So what's happening in there? So when mTOR is activated, when ketamine activates the NMDA receptor, within 30 minutes, so you see this sort of links of sausage here. So this thickening of this mTOR progenitor gene begins to turn on this whole cell growth process within 30 minutes of delivering ketamine. And then within 2 hours, these synaptic growth factors in the nucleus of the cell begin to turn on. So we can start to see how rapidly this neuroplastic process begins. So when mTOR, when ketamine blocks the glutamate receptor, we get increased mTOR signaling, we get increased synaptic cell genetic expression for the genes that control for synapse branching and differentiation, and we get this kind of final common pathway of growth. Okay, so let's circle back to this now that you have a better idea of what this is all about. So again, to review, the end of the day, we grow some synapses. So we begin by blocking ketamine at the GABA interneuron. That is going to keep that GABA interneuron from hitting the brakes on the presynaptic glutamate neuron. Without that brake pedal there, that presynaptic glutamate neuron is going to release glutamate into the synapse. When that happens, there's this burst of glutamate being released. Okay, this is sort of that gully washer of a storm is releasing all this glutamate. That's going to cause these AMPA receptors over here on the left side to be activated. So remember, glutamate is what activates the AMPA receptors. When they open up, sodium flows through. That begins this process of generating BDNF. BDNF is going to, we're going to come back to it in a minute, but that's going to activate these cell growth pathways. At the same time, ketamine is blocking these post-synaptic NMDA receptors, as we can see here on the right side. When we block those with ketamine, then we activate this cell growth pathway through the EFF2 kinase pathway. All of this, as you can see, is funneling down into increased synaptogenesis. Now, remember that increased BDNF that has been generated through these two processes? That's going to start activating these TrkB receptors right here. And you can see that begins a chain of events that leads to mTOR being activated. And as you recall, mTOR is the final common pathway before synaptogenesis. And finally, over here on the far left, we've got those extrasynaptic NMDA receptors. These ones are kind of bad news, right? Because when they are present, they hit the brakes on the mTOR process. When the brakes are hit on the mTOR process, we get less of that neural growth, that synaptogenesis. And so by ketamine blocking them, that prevents that inhibition from happening. I realize that's a bit of a double negative, but you're keeping those extrasynaptic receptors from hitting the brakes on mTOR. So all this is going to funnel down into this synaptogenic process, and we're going to get this growing of neurons and synapses. All right, it's a lot. So let's talk about how we do this clinically. So the great thing about ketamine is that there's a lot of variability in how we can use it. It's very flexible. So not only can we use it in the ER and in the emergency and in the operating room, we can also use it in various forms in psychiatry. So there are ketamine infusion clinics. They're often run by anesthesiology, but not always, where it's really just a drug-only model. So you come in, you get an IV started, you give the person 0.5 milligram of ketamine per kilogram of body weight over 40 minutes. They usually relax in a chair, but there's no psychotherapy before, after, or during. And so the patient is really just sort of passively receiving the medication and getting the biological effects of the drug. And there's nothing wrong with that. It may just not be fully utilizing the full potential of ketamine. There are a couple different psychotherapeutic models of doing this. So one could be thought of as a psychotherapeutic, or sometimes referred to as a psycholytic approach. So similar to sort of a psychoanalysis, so if you will. So using the drug in lower doses to reduce somebody's defenses in such a way that they can still talk and still have a therapeutic process. So this is sometimes done with lower sublingual doses or lower IM doses in a context of psychotherapy. So there would be some intention setting before the drug is administered. That person would probably stay with the person through the entire dosing. There might be some conversation, and then they'll talk about the experience afterwards. So that's a psycholytic or psychotherapeutic model. The psychedelic psychotherapeutic model uses a larger dose. So similar idea, there's non-drug preparation, there's the same person sitting with you during the drug administration, and the same person is going to talk with you after the drug has worn off. But the dose is probably going to be higher. It might be administered intramuscularly to make for more reliable absorption. And it's typically a dose that's high enough that it's not going to render the person unconscious, but it's probably going to make conversation quite difficult. So there's a more of an inward directed focus in that kind of model. I want to talk a little bit about S-ketamine in a moment, but just want to remind you, going back to enantiomers that we've talked about before, that when we talk about ketamine, it's typically the racemic mixture, which is the right and the left uh, side of the molecule. So S-ketamine, which is delivered in a nasal spray, is just the left enantiomer of ketamine. And there are a bunch of different ways that we can administer ketamine or S-ketamine. S-ketamine in the US is only available as a nasal spray. In Europe, it is actually available as an injection, but we don't have that here. A lot of the ketamine clinics that I'm familiar with use sublingual versions of this, or some of the at-home ketamine clinics, which I have some mixed feelings about, will use sublingual compounded ketamine. So there isn't a commercial product of compounded of sublingual ketamine. You have to go to a compounding pharmacy, have them make that for you, and it's kept in the mouth until it dissolves, and then you allow the spit to kind of stay in for like 10 minutes or so, and then spit that out, because sometimes that can contribute to gastric irritation if you swallow it. But the ketamine is absorbed through the oral mucosa, gives you about 25 to 40% bioavailability or absorption, whereas IV is obviously 100% absorption, and IM is pretty darn close to that. And so the other thing that varies is how quickly the drug will come on board. Now, S-ketamine came out a number of years ago as the drug Spravato. It's a nasal spray. It is just the left enantiomer of ketamine, so it really has the same mechanism of action as we've talked about up till now. Comes in two dosing strengths, so 56 and 84 milligrams. Um, and the way it's typically administered is a couple of times per week for the first several weeks, and then going into a maintenance phase if the person has responded. Obviously, if they're not responding, it doesn't make sense to continue to pursue this. But then you have some flexibility in the dosing between either a 56 or an 84 milligram dose. And the FDA approval of this was either for depression in conjunction with an oral antidepressant, or for rapid suicidal ideation alleviation. So that's another interesting thing, that ketamine has this anti-suicidal property. The data for S-ketamine was, obviously good enough to get it through the FDA. There's some debate as to whether or not the S enantiomer or the R enantiomer is more antidepressant. Ketamine is interesting because both of the enantiomers appear to have antidepressant properties, and some of the metabolites, such as hydronorketamine, is are also antidepressant in their own right. So taking a look at the phase 3 data here, the MADRS here, this is the primary endpoint. Anything to the left of that blue line would indicate superiority of that phase, that arm of the study. And then looking at these secondary endpoints. So this is a measure of functional disability, and this you're familiar with as a patient self-report measure of depression. S-ketamine does have a REMS program, which stands for Risk Evaluation and Mitigation Strategy. And now these are additional rules that are imposed by the FDA on top of the prescribing guidance and the label of the drug. So in the case of S-ketamine, it says that you have to keep the patient there for 2 hours. It's never a take-home drug. So this is why, ketamine, racemic ketamine, does not have a REMS, because all of its use in depression is off-label. And so interestingly, S-ketamine has more regulations around it than racemic ketamine. And some of these things include, like, you can't take it home. You have to stay at the clinic for a couple hours after it's administered, etc. So any future psychedelic treatment will probably require a REMS program. It just remains to be seen what exactly that will entail. I mentioned that ketamine has this rapidly antidepressant effect, antidepressant effect. It also has a rapidly acting anti-suicidal effect. And this was discovered quite some time ago now, about 2010 was the first reports of this. And, I think this really is something that we need to think about in terms of emergency treatment of suicidal ideation. I mean, we know that we have a suicide epidemic in this country. And what if we looked at ketamine in the same way that neurology looks at TPA for strokes? So if you could give somebody a drug that could rapidly change their suicidal ideation in the same way that giving somebody TPA who's recently began to show signs of a stroke can reverse those symptoms. What if we could do that with ketamine for depression? I think really it's an opportunity that deserves to be further explored. Now, the costs of ketamine and esketamine are a barrier. Ketamine itself, racemic, it's generic, it's cheap, but often what drives up the cost to several $100s per infusion is the amount of time that it takes. So you've got somebody sitting with a patient for a couple of hours. You have to pay that person. And so we often see in the Bay Area, at least, that ketamine clinics are charging $500 to $1,000 per dose, which is obviously a big barrier for many people, given that ketamine is considered experimental by a lot of insurers and therefore not covered. S-ketamine is FDA approved and covered by a number of insurers, but is more expensive per dose. So the drug itself can be several $100s. Now, what about a ketamine-like drug that can be taken orally? And turns out we actually have one of these, and we've had one for a long time. It's called dextromethorphan. And so dextromethorphan, you might be thinking, I've heard of that. And it's a cough medicine, right? It's a cough suppressant. The thing about dextromethorphan, it's a glutamate receptor antagonist, like ketamine, but it's a very short-acting one. And so it doesn't really stick around in the body long enough to cause these sort of neuroplastic effects unless you inhibit its metabolism. And it is metabolized through cytochrome P450 2D6. So what if we combined dextromethorphan with a strong CYP2D6 inhibitor? And that's exactly what bupropion is. Bupropion also happens to be an antidepressant, as you know. And so the idea here was that we give dextromethorphan and bupropion twice a day. The bupropion inhibits CYP2D6, keeps dextromethorphan around longer, and in the studies that got it FDA approval, found that it had this rapidly acting antidepressant effect. Not quite as quick as ketamine, but within a couple of weeks, you had almost 50% of the dextromethorphan group remitting, not just responding, but remitting, versus about 16% of the bupropion group alone without the dextromethorphan. Now, what's interesting about this is that both these drugs separately are readily available, generic and cheap. So, you can go on Amazon and get 200 tablets of 15 milligram dextromethorphan. So you would need, let's see, you'd need 6 of those a day. So we're looking at about 30 cents a day. You can get that bottle for $10. Bupropion, 100 milligrams sustained release, which you could take twice a day, is about $14. Or you could pay $1,300 a month for the brand name of this drug. I'll let you figure that one out on your own. So we've talked about the NMDA receptor primarily as a primary target for these glutamatergic treatments. But what about mGlu or AMPA? And it turns out there is research into this. So mGlu at 2 and 3 and 5 have been looked at for treatment of schizophrenia. And just recently, there was some data announced that there's a drug that would be a positive allosteric modulator for AMPA, which has just gotten through phase 2 studies with some positive findings. So we may find that down the road, there are some additional treatments that would be available using the glutamate system that don't necessarily target NMDA. And so, a lot of times when we talk about ketamine, we talk about it being sort of reserved for treatment-resistant depression, which seems to be where it's kind of fitting into our treatment practices. We don't really have a contemporary STAR-D to give us guidance on this, but this has really given me a different kind of hope for our patients who've had difficult to treat depression. And we've often called that treatment-resistant depression after they've not responded to two, usually the guideline is two different types of antidepressant medications. So like an SSRI and a norepinephrine reuptake inhibitor have not responded. And so we would call that person having treatment-resistant depression. But I wonder if it might be more accurate to call it monoamine unresponsive depression. So when we're trying to tickle norepinephrine or serotonin, we don't get a benefit. But maybe if we were able to go to a glutamatergic drug like ketamine or esketamine or dextromethorphan, then we could get that response. And we don't really have clear guidance as to when and how to do this. So this is an interesting recent paper, just kind of reminding people of these response rates in some of these procedural treatments, such as TMS and esketamine and ketamine, hover around 50%, which is several times better than many of the third generation antidepressants or augmenting with a second generation antipsychotic. And so it really begs this question of like, when should we begin to think about using something like ketamine or TMS in our treatment sequences? And is there a cost of allowing somebody to stay depressed for a long period of time? And, I think the answer to that is yes, because the brain gets good at whatever the brain does a lot of. So we don't really want a brain staying in a depressed state for a long period of time because possibly it makes it that much harder to treat. And so if we can get somebody out of depression and get them responding sooner rather than later, that probably foretells a better course of that person's illness over the course of their life. So I think we're still sort of trying to figure this out, but I think it's going to be your generation of clinicians that are really moving into this different paradigm for treating depression. So thank you for your attention, and stay tuned for the next part where we'll talk about GABA and its use in both depression and anxiety. Thanks for your attention.